Pwede
Okay.
go.
All right. Welcome to Youth Talks on ICANX, Young Scientists Shaping the Future. My name is Martin Thuo. I'm coming to you from the Eastern United States, from North Carolina State University. I'll be moderating today. And our speakers today are going to be Professor Charlie Mace and Professor Bharat Gwalani. And our panelist is going to be myself and Alice. Our first speaker is Professor Charlie Mace from Tufts University. Charlie is an associate professor in the Department of Chemistry. He earned his BS in physics, and followed by an MS and a PhD in biophysics from University of Rochester. Then from there, he moved to Harvard University to work with Professor George Whitesides as a postdoctoral fellow. Prior to joining Tufts, he joined Diagnostics for All, a nonprofit company with a mission to develop low-cost devices that reduce global disparities in access to diagnostics. At Tufts, Professor Mace has carried out a fantastic work focusing on developing simple yet innovative medical devices that will lead to increased access to healthcare. Their efforts aim to bridge technology development with clinical applications, and their approach includes point-of-care diagnostics and patient-centric microsampling devices designed using paper-based microfluidics. Charlie has won many awards. Among these is the Emerging Investigator by Analytical Methods. He was named a Rising Star and Top Expert by Analytical and Bioanalytical Chemistry, and he's the recipient of the NSF Career Development Award. Charlie, I don't want to take more of your time. Please share your slides and take it over.
Excellent. Thank you for the intro, Martin. I really appreciate it. I appreciate the invitation. This is really an amazing series to be a part of, and I'm glad to have the opportunity to kind of walk through a little bit what my research group does. And so, this is, I've seen a lot of talks through the ICANX series, and a lot of really amazing, sort of cutting edge, innovative research science. And I want to get into that my group does as well, but I wanted to also spend a lot of my time today really introducing more of our methodology, not just the experimental methodology, but sort of our thought process around how we even jump into a research project and where that's led us. And so sort of by design, really intentionally, I'll be introducing some really early stage results from the group, things that will hopefully be, published in the next year or so. And so it's not just about device prototyping as being generally an important part of translational research, but it'll be from our perspective being, as Martin introduced, paper-based microfluidics, paper diagnostics, trying to impact point of care. And that's really the whole purpose of this is impact. And so as Martin, who's a colleague of mine at Harvard, we overlapped as postdocs. There's a prominent scientist once wrote, a perspective about how to communicate your research, how to publish. And one of the big questions here is always, who cares? And that's one of the biggest lessons that I took was not just that you need to sort of justify what you're working on, but it becomes a critical part of even the experimental design process. Like who cares about what I'm doing? What is the purpose of my research? What is the kind of impact? Who benefits from this? What does success look like? And what I want to try to get through is that success can be quantified, by having performance metrics that you sort of define in advance. You can point to them and say, look, we did the thing that we're supposed to do. We did the thing that we set out to do. And that can be a very powerful and unbiased way of looking at success in your research program. But this all needs to be addressed very clearly, articulated very clearly, and as accurately as possible. Because when we think about this big question of who cares, at least in my space, which is ostensibly sort of point of care diagnostics, medical devices, assay development, this image kind of encapsulates what that means. It's real world impact. And this is something that we as scientists, as reviewers, people that are involved in funding agencies, just the general public, we want the promise of some kind of impact from the basic scientific research that's happening. So this here, what I'm pointing at here, like this is tangible impact. This is the picture here is a clinician performing a rapid diagnostic test in the field to determine if a patient has lung plague, so Yersinia pestis, sort of colonizing the lungs. And so this had to be developed somewhere. This chemistry, the device that they're using, the kit had to be packaged somewhere. The instructions needed to be developed in a way that this person needed to be able to accurately and correctly use that assay and get the right determination. This is really, really difficult. And for academic research in this space, again, point of care diagnostics, medical devices, this kind of impact can be really hard to support, because this stage where you're actually impacting a real patient's life is difficult to achieve. There's just too many obstacles between sort of what we can do in an academic environment, but also just from the perspective that we're not here to make products. We're here to train students and to teach and to innovate and de-risk new ideas. But that being said, every abstract introduction conclusion is going to have something that reads like, we detected this marker to diagnose this disease with a device that has these attributes to improve outcomes in this population. And we've seen that a lot in the last couple of years. One of the reasons why we're interacting by Zoom is that this became one of the most prevalent ways to communicate during sort of the height of the COVID-19 pandemic, where you saw a lot of manuscripts that would come out, us included, as talking about their SARS-CoV-2 test, whether it's a molecular test or an antigen-based test, and justifying why they performed the research the way that they did to improve limits of detection, lowering costs, increasing access to home testing, but a lot with qualifications of whether they're explicitly stated or just sort of implicit, because again, it's academic literature, that if the star is aligned appropriately, the groundwork, the foundation that we've laid today could eventually have an impact in the way that we've talked about. And so if you've done this sort of correctly, this communication correctly, it shows you have a vision and that you're on that path, and that you can sort of in a very objective way describe your contribution to that path. When done incorrectly, it looks very much like overselling. And that's, I think, a very dangerous place for academic researchers to be, sort of oversell what they've done. And so it's a very fine line to walk, but it's one that we all do, right? So I'm not going to sit here and saying that I'm not hypocritical. This is how we talk about our science as well. And so, one of the very first papers out of the group, we developed a device to perform immunoassays, and it looks something like this. And I'll show little examples, here and there, as long as my screen doesn't pop out at me. It's a little paper device, multiple layers, that performs similarly to a lateral flow test. But how we pitched it in the introduction, I've highlighted that here, is that our results suggest that paper devices may find broad utility in the development of immunoassays at the point of care. That's not what this manuscript was about. This manuscript was technology development. We compared results using not contrived samples, and now actually, a low number of sort of patient-derived samples. We compared it directly to a clinical to a commercial product, and showed that it, n equals 2 or 3 or so, that it performed comparably. In the next one, a few years later, this is about measuring hematological parameters, so hematocrit, the ratio of red blood cells to overall volume of your blood. We say ultimately developed an inexpensive, 3 US cents per device, where we could determine the hematocrit, and the results provide a foundation for the design of other devices that enable separation detection of cells in limited resource settings. Both of these kind of have the same flavor of point of care, limited resource settings, lower cost, because that's the intent. That's where we would like this technology to go. That's where we think technology like this would fit the best. But we weren't using this to the point of care. It was in our lab. We weren't in a limited resource setting. It was in our lab, which is ostensibly a pretty high resource setting, actually. And when it comes to the cost here, 3 cents seems really inexpensive, but that's just the bill of materials for the paper and adhesives that we used to build the device. That's not a real price to an eventual consumer. Maybe it's going to be higher because we're not introducing things like infrastructure costs and equipment costs and salaries and all the other things that go into making a product, but maybe it's lower because now we're operating at the economies of scale that sort of drive costs down. We have no idea. And so some of this is really forecasting, some of it's wish casting, like we hope it's going to turn out to be something like this, but it requires a very sort of sober and long-term view on the work. And we went into these projects not necessarily with a target in mind, just that we wanted to develop technology. But sometimes you want to go in with a target in mind, whether that's a target that you define with the help of others, or the target that's defined for you by the World Health Organization, Centers for Disease Control, et cetera. And so for something here, and this is not a real target product profile, but it's an example of some of the things that we seek out, is someone giving us a goal, like for example here, a quantitative test for HIV viral load you can perform in the home. We want this to be specific for HIV-1. Ideally, we're targeting a conserved region of HIV-1 to cover a lot of subgroups that you'd find out in the field. We'd like to have a limit detection that is consistent with early diagnosis, or at least something that can be used potentially to monitor the effect of antiretroviral therapies. And from an accessibility perspective, we actually can quantify something like that. Like, how easy is this going to be for someone to use? We would do that through focus groups, through user experience research, things of that nature. But all of these, from specificity, from limits of detection, through usability, these all end up being, from my perspective, I think, directly in the academic sphere. This is research, this is development, this is learning, this is innovation. A big part of technology development is technology use. It's not just the function of what you made, it's the form of what you made. And I think function and form go together hand in hand in a really important way, particularly as we kind of think back to a few slides ago where we're talking about impact, right? Someone eventually needs to be able to use this, whatever you've developed. And so linking form and function together, or at least not necessarily prioritizing one to the detriment of the other, becomes a critical aspect of your experimental design and putting it all together. And so again, if we're trying to be really quantitative about this, and sometimes we have metrics like this and other times we don't, we'd like to be able to have something we can point to and go, we've been successful here. We've made the step that we needed to drive our technology forward. And so the best tool that I've been able to find so far is called the Guidance Impact Tracking System, GATES. It's from a local group sort of in the Boston area through Mass General Hospital and some others. It's the Consortia for Improving Medicine with Innovation and Technology. It's called CIMMIT. I'm not affiliated with these groups, but I've had the opportunity to use this platform before. It's this really nice wheel that directly outlines all of the steps that one would need to take on a technological side, on a regulatory side, marketing and business, and then clinical, to go from the foundational aspect of your research, which is the overall need. Why are you even doing this? Who needs this? All the way around towards it being the standard of care. And so I'm not focusing at all on regulatory and market and business. I could give many other talks on this. I want to highlight the technology path today and then kind of bounce back and forth to the clinical side as needed. And so I'm gonna blow this wheel up a little bit so we can see things better. And so this idea of kind of starting off at need and an idea, don't worry about what's filled in here. This is just demonstrative from their website. The need and idea is really what we would kind of consider to be the preparatory stages of a standard research project. We're doing literature reviews in order to understand what the problem is and what's been done to address that problem and why there are gaps that need to be filled. And then in the idea stage, it's rigorous experimental design, trying to figure out what you're going to make, how you're going to make it, if it's what materials, reagents, approaches you're going to use, et cetera. So we're as academics, we're very well versed in sort of traveling through these stages. And so this feels very familiar. This feels very much like home. And then proof of concept, these are your first prototypes. They could be crude, they could be something you've been working on for a decade, but these are really things that you're making with your hands. They're allowing you to reduce your idea to practice. You can show off key aspects of performance, using contrived samples, spent patient samples, getting limits of detection, quantification, generating calibration curves, determining sensitivity and specificity. This ends up being part of the applied aspect of the research program, showing your device work, showing your assay work, showing it has the performance that you would like to have based off of the earlier stages, which is the need back in the idea. And then we move into proof of feasibility here. And this is where sort of the wheels fall off a bit from this overall wheel. This stage isn't iterate or improve. This stage isn't use more samples. This stage isn't even necessarily pilot your device or your assay in a field setting. This is kind of freezing your design overall and stopping doing research and start doing development, identifying places this can fail, identifying manufacturing pathways, which means not your hands anymore. This is a third party that's semi-automating or automating at either batch pilot volumes or leaning towards higher volume production runs, negotiating deals with vendors to reduce prices. We don't do that, right? At least at Tufts, we have a university negotiated rate for materials. I'm not doing that independently. So again, like we don't really do this. We can do some of it, but we can't do it all. But if we're trying to eventually give what we've made to others to evaluate its potential, then we kind of have to do some of this. We need to have some confidence in some aspect of the performance that are with our partners. Otherwise, we're risking time and resources. We're risking turning our partners off. Like, well, this works for me is a really bad response to something failing at the pilot stage. And trust me, it feels really, really bad when you've sort of made this implicit promise that, like, yes, I'm going to send you devices. Please use this. Please use your time, your resources, your efforts, enroll your subjects, your donors in this, and then everything kind of fails. It feels really bad. So this is a bridge almost too far for us in a really difficult way, because what that means is we never can address any other steps, proof of value, clinical trials, validation, all the way through. I'm talking about approval and launch. This is not selling millions of units. This is having anything that looks even remotely commercially viable, right? This is the only place where impact can really happen. You need to have some approval process that says, yes, you can sell this device. Yes, you can sell this assay. We've addressed all of the risk associated that could be, let alone getting all the way to standard of care. There's just a really long way between where sort of we traditionally stop as academics and where impact begins, let alone, again, this widespread adoption. And so this isn't necessarily to say like we should throw our hands up and never try to look forward, very future forward into what would be needed. Because again, if we're trying to pitch this idea of adoption, of impact, of helping the real world, not just developing new science and publishing, which of course there's nothing wrong with that. That's what we do. But I think there's a lot to learn from understanding what other people are going to be looking for in your technology and building ways to address that into your own research programs. And so that's looking to sort of where we're putting our money, where our mouth is. This is going to look like a standard slide of like, look how productive we've been, look at all these things. But what I really want to show is sort of the history of the group in going from more of a diagnostic assay development workspace, kind of all the way through the patient-centric microsampling devices that Martin had mentioned in his introduction. Because again, the goal for my group, my own personal goal as a scientist has been to actually make something that someone else uses, and that's a really important thing for me. That's my white whale, is my Ahab being maniacal going after this goal. So we started off with immunoassays, right? This is one of the very first things that I showed up. Eventually, this would need to be an in vitro diagnostic, going through the FDA's route or going through the European Union CE marking route. This is a really long, expensive road. And we've done a lot of really nice research here that others in the space have done also really amazing work here at Hadley Sykes at MIT and others. And I was enthusiastic about this at the start of my career, but became really clear early on that we were going to be reagent limited. Building our assays, we would either need to also be reagent developers, which we are not. We don't have that proficiency, or we'd need to only be able to rely on things we could buy commercially. That's ended up not feeling really good as a place to be. That meant to me that we'd be really locked into this as a platform development tool. And we'd eventually need to very much hand this off to other people in a way that we wouldn't be as big of a part of. So we went to bigger problems that weren't being even addressed by others, much more early stage sort of in the need, which is in hematology. There are very few solutions out there for looking at parameters of your blood cells, right? So again, this is our hematocrit device. where it was this thermometer style readout where the longer the distance the blood fills your channel, that means there's more fluid, more plasma volume, fewer cells, so a lower hematocrit, less fluid, higher hematocrit, more cells, and try to get a nice visual guide between where you're in a normal range and where sort of clinically low and clinically high. And this ended up building us off into other areas. We started to understand sort of the role of a porous medium and how it either restricted or enabled the transport of cells. These sort of, think of particle flow in porous media, big squishy, biconcave disc red blood cells behave differently than sort of bigger and spherical and more stiff white blood cells. But this gave us some really interesting intuition into doing some material science, doing some assay development, doing some device design to be able to build a device that can detect white blood cells. We really, really like this. But again, like this is really good basic science research. This is very, very far away from clinical real world impact. But that there was still that itch that I had, which is to get something into the hands of others as fast as possible. And so when we looked to that space, it ended up being in specimen collection, dried blood spot cards, dried plasma spot cards, where all of the testing apparatus is still back in a clinical laboratory. We're not developing new assays. We're innovating around how the blood is collected and shipped back to a lab. That seemed to be something that would be faster to getting into the field, making a device. There's not a lot of chemistry in this. This is more engineering at this stage. But the engineering aspect needed to support the chemistry in a lab, whether that was more of a high touch assay that was being developed or an automated assay, et cetera. This is really where we want it to be. And so this ends up being more the focus of the rest of my story here is on our pathway into developing these dried blood and dried plasma spot cards. So if we're looking at a traditional Wattman 903 style unpatterned dried blood spot card, this is sort of the Hollywood perfect blood spot, right? We're pipetting blood directly onto these circles. These circles are just toner. They don't do anything. They're just there to guide the eye. This is sort of a horrible looking dried blood spot card. We sort of made this in a contrived way to show where this could fail. This actually looks very similar to some of the failure modes that you see in the literature for dried blood spot cards. Either the spots are too small, they're smeared, or they bridge. And so if the point here is to be able to extract a sample, make a measurement off of what is being stored there, from an analytical chemist perspective, I want to know what my volume is. I want to know how much of my what the starting sample is there, because I want to tie that back to the state of the blood at the time when it was drawn from the finger stick. And if I'm not getting matching there, if I have no concordance between sort of liquid blood and my dried blood, then what's the purpose of this, right? So really what we had to strive to do was to make this match as best as we possibly could. And so this became a design challenge. And so, here's some data kind of showing again, different unpatterned dried blood spot cards just with circles by eye. We're spotting blood down by pipette at different hematocrits, pathologically low to pathologically high. And here's what it looks like in our cards. And so we've constrained where blood can flow. So these patterns are now active. They're making channels. They're now our paper microfluidic devices. And not only are we able to constrain where blood is flowing, but if we're trying to extract a specimen after the fact, we now know exactly where to extract the sample. There's no guessing. There's no lost sample here. You see this punch over here. You're getting a lot of paper with no blood in it. That's a big challenge. That means you're undersampling there. You have a lower response from your measurement. We don't want that. And so we have now something that fills really uniformly. We have consistent placement. We have metered volumes, and metered volumes is really enabling, because we can know if I know there's always 10 microliters in this punch, not 8, not 12, not 6, not 15, always 10. I can start to improve the accuracy of that measurement, again, back to what would be in a more of a liquid blood sample. And so not only do we get better measurements, we can actually get new measurements. And so in comparison, so here's a standard dried blood spot card taking 3 sub punches from there. And this is the volume that we've measured that's contained each of those punches. Each of these colors corresponds to a different card, and then sort of replicates within the card. You see just a massive spread. And so if we do a statistical analysis between the volumes we collect, both within the card, intra-card, and then across cards, we have no consistency. We have, the volume differences within and in between are statistically significant. Whereas with ours, we're sampling more in that same size of punch, a much larger volume, and with a much more increased precision. And so we feel really good about saying that this card design, at least as we're making them manually still, this card design gives us this volume of blood in every single one of your punches. What that can do in a way that a dried blood spot card to date has not been able to support is the measurement of an accurate white blood cell count. And so by quantitative PCR, we can amplify a white blood cell gene that, back to a calibration curve, that gives us an absolute number of white blood cells, but the count requires a volume determinant. And so by knowing how much volume there is in that punch, we can go from an absolute count in terms of magnitude down to sort of more of a concentration in terms of white blood cells per microliter. And now I'm only showing a little bit of data in this talk, so I don't want to focus on the data. I want to focus again more on the methodology. We're getting really decent accuracies back to what the standard ends up being. And some of the errors are a little bit larger than we'd like them to be, on the order of 20%. But at higher counts, our accuracy is our error is 3%. This is a really highly accurate count off of a dried specimen, not off of a liquid specimen. And so to kind of circle this back to the beginning, again, with this question of who cares, right? So at this point, we're just kind of talking about platform development and sort of demonstrations of that platform. But one of the areas where people really care about microsampling is in understanding transmission and drug resistance in malaria. There's some sample processing, sample preparation, sample stabilization that's required in order to do genotyping on malaria parasites, which are stored within red blood cells. And then the other one is collecting metered dried plasma. There are a bunch of plasma collection cards on the market, but getting the precise reproducible volume has been a real challenge. And we think that with the technology, the approaches that we've developed, we can meet this goal. And then the next question is, where does focusing on prototyping benefit the goal of the research? That's the title of the talk, right? I haven't really talked much about prototyping yet, but the goal here is to support field research, support impact, support evaluating the applicability and the performance of the technology in hands that aren't yours. And then from an analytical perspective, decouple the error that you get in the measurement from error in assembly, right? I want the error to be in how this was measured, standard measurement error, standard assay error, and not from misaligning things. And then how far can we get back in that innovation cycle? They're probably still to the border of proof of feasibility and value, but by doing it in a more direct way, the hope is that we de-risk what could be required in the future and make that transition through the cycle a little more realistic and reasonable. So I didn't focus at all on the clinical wheel, kind of purposefully, because what's in this sets up what we're doing next, which is talking to people, right? Not using the scientific literature as a basis for what you do, but using people as the basis for what you do. All of these stages here are basically talk to more people, talk to newer people, generate information, get feedback on the form of the device that you've made. The function should come, but the form of it, make sure that it's going to fit in with their workflow. Because if it doesn't, they won't use it. If they have to completely change how they do and what they do, it's very unlikely that you're going to get any kind of adoption, and that leaves you sort of dead in the water, which is not necessarily the overall goal of the research program. So here's an example, right? So we have our malaria device. This is going to operate by allowing parasitized red blood cells to move down one of our panels while retaining white blood cells in a different area. So rather than having all of that genetic information mixed, have an enrichment of the parasite genetic information by depleting the white blood cell human host genetic information. So this started off around 2021. We had a first device sent to our collaborator in the US in Maryland. Got some feedback, changed the materials, changed some of the patterning. That got sent to Ghana for some pilot field studies, got some results back there. Again, changed the form, changed the material, started adding, we've added now these arrows to try to help guide people in the field in terms of how they use them. That testing data then informed a bunch of different designs, which then got sent to Peru for a different field site. We're kind of chasing when malaria seasons were endemic. That feedback changed again. So again, what we're seeing is like we're talking to people, we're doing some pilot studies, we're getting information back, the function of the device isn't changing. Its form is changing. It's tiled across. We have more inputs. There's more space for adding patient information. It's a bigger card, so the people in the field can handle it better. This fits into more standard drying racks they already had, because now it's the exact same footprint as the Wattman 903 cards they had been using. And again, kind of all the way through. And so we're starting to get, we've gone from getting poor data to getting really exciting data. And it's through talking with people that are using devices like this, and then also this in real field settings. Similar to the dried plasma spot card, we kind of developed this in a vacuum, where the idea was you'd apply a sample of whole blood to a filter stack that would remove all the cells, and then the plasma would be pulled vertically and then laterally to collect in these little satellite zones. And we could, with this pure plasma, have multiple assays can run. We can go back in the middle with the blood punch, sub-assay that for any sort of host genetic information that's there. And again, really interesting research development, but not where we want it to be. And so we want to enable field testing. We want people to use this. We want to help the work that's being done clinically that's relying on the collection of a dried sample. So our first generation prototype had 75 microliters of blood in. The output of plasma was okay, so sort of 2 8 microliter zones. We looked at the overall testing menu of what you would want to use for plasma, not whole blood. HIV became the most prominent thing we wanted to address, HIV viral load in particular. And so when we talked with our partners, we said, ideally, they don't want to punch, they want more plasma volume, and they want to be more efficient. And so this led to our second generation device, pre-perforated zones. We're adjusting device dimensions. We're considering zone geometry, everything else, right? So 2.5 years of research went into just iterating prototypes, not even necessarily checking if they worked yet or not, getting feedback on form from our partners, and then showing what the function actually is, and doing it in a way where we could make a ton of these. Beautiful glamour shot. We made a few hundred of them, packaged them in a box, and then sent them off to the National Institute of Communicable Diseases in Johannesburg, South Africa, to be evaluated in their clinical setting. And so between the malaria study and then this HIV viral load study, I think end of 23, beginning of 24, you should start to see sort of the fruits of this labor, which ostensibly is prototyping for form and function. We don't want to stop at proof of concept. We want to talk to key stakeholders and collaborate to iterate, not around how it's supposed to work, but about how it does. Bridging key gaps in manufacturing. So again, we're taking away that kind of error from how we're assembling manually in the lab, semi-automating to the best of our ability, because again, we're not a manufacturing house. We want to kind of avoid these sort of gross errors that come from making things in small batches. And so our batch process allows us to make the few hundred devices at a time that our field partners, any other collaborator would want. And then also instructions for use. How do you communicate your work? It's not just in a written form in a manuscript, kind of answering that who cares and why you did it question, but being able to communicate your methods through an instructions for use, both in words and visually in a video format, to try to, again, a different kind of error, which is a gross usability error that someone is just misusing. They're spotting blood in the wrong spot. They're disassembling it incorrectly, et cetera. How to get things that work in your hands really well because you're the expert and you've optimized it, getting it to work in the hands of others. And by others, I mean absolutely not me. Right. One of the best parts of this job is that I get to come and speak with folks like you and participate in this conference series. But it's really all of these folks, the current and former members of the lab, all of our collaborators and funders that have really allowed us to have the space and the capability to identify what we think are really important needs, where real impact can be made, and then do the basic research development, but then also the development that's towards translational work. Through prototyping, through manufacturability, through form and function. I'm looking forward to participating in the panel afterwards. Thanks so much for the time.
Thank you so much, Charlie. That's very, very exciting. Great work that you're doing there. It's my great pleasure now to welcome our second speaker, Professor Bharat Gwalani. Bharat is a colleague at North Carolina, and a little bit about him. He's an assistant professor in material science and engineering at North Carolina State. He receives his Bachelor's of Engineering from the National Institute of Technology in Jaipur, India, and then went on to obtain a PhD at the University of North Texas, both in material science and engineering. After that, he went up north to Pacific Northwest National Laboratory, where he was a senior material scientist. And then after that, he came back to the Southeast to start his independent career. His research focuses on mechanistic understanding of materials vulnerability under extreme environments, deformation-assisted modification or phase transformation, and the development of characterization of advanced materials. Bharat, the floor is yours.
Okay, thank you, Martin. Thank you for the introduction. I would share my screen. Let me know if you can see my screen.
Yep, it's good.
Okay, perfect. Okay, good morning, good afternoon, good evening, everyone. I know the audience are spread across the world. And thank you, Martin, Professor Martin Thuo and Alice for inviting me over here. I feel privileged to give a talk to such a wide audience. As Martin mentioned, I am an assistant professor at University of North Carolina State University, and I recently joined here as assistant professor. I worked in Pacific Northwest National Lab before joining here. Today, I'm going to talk about deformation-induced modification of phase transformation pathways in metallic systems. This has been core of my work for last 5 to 10 years, this particular topic. And so I try to summarize and also talk about some generalities in different works that I have done so far. Okay. Before I talk about my research, I just wanted to talk quickly about where I come from. Let me try to see if I can point out. So I come from a city called Jaipur in India, which is northwest of India. And the city is pretty old and historic. It's known for its palaces and its history. It has most of the historical buildings made out of pink color because the sandstone available was pink. So it's called the Pink City of India. I just wanted to shout out for the city. It's a beautiful city to visit. So please feel free to visit whenever you come to India. Okay, I did my bachelor's in metallurgical and materials engineering from National Institute of Technology, and I have some experience in working in steel industry. I worked in stainless steel for about a year and then mild steel for 2 years after that. So that experience was important for me to go towards a doctorate degree because it allowed me to understand and see material in its real form and then create that curiosity and those questions that why does material behave differently? What does this different colors mean? What are we doing in terms of chemistry when we are trying to reduce ore to a shiny metal? What are the different types of defects that come in the metal? And how these defects influence the properties. So all those things were seen over there, but there was a missing gap in my understanding from the atomic scale. And that's where I thought I need to look at the material at a higher magnification and try to understand what's going in there. And so I came to US, did my PhD in developing precipitation strengthened high entropy alloys. I'll talk what high entropy alloys are a little bit, and I'll introduce what my research was during my PhD. Then I went for postdoctoral research did some postdoctoral research at University of North Texas, and then went to Pacific Northwest National Lab. I'll talk a little bit about my work at Pacific Northwest National Lab as well. It's a DOE, Department of Energy National Lab in US, where I did some deformation processing and understanding of microstructures in metals. Okay, this is a summary slide, which I just wanted to share in a nutshell what my work has been so far. I like to understand phase transformation pathways and try to understand the microstructure and property correlations. What I mean by that is when we provide different conditions to a metal, it goes through different microstructures and atomic arrangements that determine the properties. By properties, we can talk about mechanical properties, how strong it is, how ductile it is, or it can be degradation mechanisms. It can be so many other things. All is determined by the microstructure and atomic arrangements. So I like to understand how this microstructure influence the properties. Then I try to provide those type of different conditions, the extreme environments that a material has to go through during application in a laboratory scale, where you can provide the type of stresses and environment the material has to go through, and then try to understand locally how the microstructure changes and what is the response of material. So a lot of my work has been using site-specific in situ techniques to provide those kind of extreme environments and try to understand how microstructure responds to that. So in nutshell, I have listed 5 different points where I have been trying to make inroads, trying to make materials which can perform better, try to go through different innovative material processing routes where we can develop the material for advanced applications, try to understand material vulnerabilities, where we can understand how material responds in different extreme environments, and develop nanomechanical and site-specific deformation techniques to understand how we can see at atomic scale how material is changing its formation. Okay, quickly acknowledging the sponsors and places where I've worked. I talked about Pacific Northwest National Lab. There was solid phase processing, which funded a lot of my work, which I'll be sharing today. University of North Texas, where I did my PhD. A lot of my work was funded by Air Force Office of Scientific Research in US and Army Research Lab in US. So I just wanted to shout out for them. My team, I have a team of students at NC State, Michael and Farhan. They're working on some of the things I'll talk about. Then some of the work is from PNNL. So I just wanted to acknowledge my team from PNNL. Mayur and Anchi provided a lot of results, which I'll be talking about. Okay, let me now jump into my work. So before I start talking about some of the results, I just wanted to talk a little bit about how do materials get stronger. One of the methods that we use in material science is called precipitation hardening. What happens over here is we can introduce very fine scale precipitates or phases inside a microstructure, inside the grains, that can then make the material stronger. And one of the systems I would talk about today is aluminum copper system, where if you provide the right type of temperature and condition, you can start forming copper aluminum phases inside the microstructure. When these precipitates form, the material becomes strong. But the important thing is the size scale and the boundary that forms between the precipitate and the matrix, right? If the boundary is not coherent, if the aging time is increased, the precipitates go from a coherent interface to an incoherent interface. And then the yield strength, which is the flow stress at material starts to deform plastically, reduces. So we want to be somewhere where the precipitates in the matrix forms a coherent interface. By coherent interface, what we mean is there is a arrangement between the matrix and the precipitate, which introduces slight strain, but there is a registry between the matrix and the precipitate. Okay, so you want to form precipitates that are coherent within the matrix, which increases the strength of the material. When they become incoherent, the material loses its strength. And of course, we want stronger materials in certain conditions, right? So I'll talk about where these aluminum copper alloys are used. These are used in automotive applications. These are aluminum alloys which are lightweight. They can sustain high temperatures, and they allow higher efficiency of the engines that we use in our IC engine cars. So we want stronger materials to higher temperature, because if you want to operate an engine, the temperature goes higher. And what happens is if the material becomes soft, then we lose a lot of the structural properties and the material will fail. So this graph quickly shows that when we have these coherent precipitates, which are theta double prime, theta prime, and theta, the strength of the material falls down as we go from certain type of precipitate to an incoherent precipitate as we increase the time at a high temperature. Okay, and this is an in situ experiment which shows that these are the coherent precipitate, which has a coherent interface. And if you keep annealing at 400 degrees Celsius, it forms another phase, which is this blocky phase coming at the interface, and the material will lose its strength. Okay, so the idea over here is, if we are able to sustain the material to high temperature for longer durations, we'll make it more efficient. right? So we have to come up with a solution where we can introduce something in the material which will sustain it at a high temperature for longer durations, right? And that's what the alloy development part we did. So today I will not talk a lot about how this alloy development was done, but I'll talk about why the material became stronger at high temperature. Okay, so what we did was we in aluminum copper alloys we added small amount of manganese and zirconium. Okay, and the observation was as we change certain compositions, we add magnesium, we add zirconium, we add manganese, we add manganese and zirconium, and we change some amount of copper. This is how the material performs. This is the hardness, which is similar to the strength of material, and this is the temperature. As you are increasing the temperature in a case which is this one, we just add magnesium, the strength falls down about 200 degrees Celsius. When we add certain other elements like zirconium, no zirconium or manganese, the strength falls down. So that means the application temperature cannot be higher than 200 degrees Celsius, right? The strength falls down to the material. But when we add this manganese and zirconium together, we start seeing that the strength is maintained. So this was very curious. We figured out a way where we add some amount of manganese and zirconium in the material and the strength of the material is retained. Then why is this happening? So when we started looking at the microstructures in scanning electron microscope, we see that these precipitates, which are these elongated phases, if they are made to be stable till high temperature and they don't convert into a incoherent phase, then material remains to be strong. So there has to be something to do with these precipitates being stable at high temperature. And that's what we see. In a microstructure where manganese and zirconium are added, the high density of these elongated precipitates is found even at high temperature. So then the question becomes why manganese and zirconium are able to retain the stability of these strengthening phase. Okay, let's try to again look at it once more. What we are doing is we found that up to 350 degrees Celsius, these phases are stable, right? We are able to see that these precipitates are stable. But let's see what happens at high temperature. Just to come back to a lower temperature later, we just started going to a higher temperature in an in situ framework. We are trying to anneal the microstructure at 400. We start seeing that at the interface of the matrix and these elongated phases, these blocky precipitates are precipitating, right? So this is dissolving these elongated phase and a new phase is coming up, which is not desirable, by the way. At 400, this is happening. But till 300 and 350, when we do these in situ experiments, this was not happening, right? And we are not able to see anything else happening, but something must be happening, right? Because these things are stable. And when you don't add manganese or zirconium, these things are not stable, right? So why is this blocky phase not forming at 300 and 350 degrees Celsius? Okay, so that becomes a question and we were not sure why this is happening. So then we designed an experiment to understand what is happening at 300 and 350 degrees Celsius and why these things become stable at lower temperature. So over here, I just wanted to talk about a specific microscopy technique that I use to solve this problem. This is called atom probe tomography. Okay. Over here, we make very sharp needles. These sharp needles, by sharpness, I mean they are really sharp. They are 50 to 100 nanometers in thickness. As you go, this is a conical needles. At the tip, they can be up to 10 nanometers actually, and you have to make it really sharp so that when you take it in a very high voltage, the field of this pulsating voltage can start evaporating material atom by atom, plane by plane, and excite it towards the detector. Okay, and then this detector, from the time of flight, position where it is being collected, and the z coordinate, is able to recreate the reconstruction, talking about the compositional change as the material is being evaporated. So it's a destructive technique, but you're able to recreate the material in a software and talk about the composition of it. Okay, so we use this technique in solving this problem. And how did we apply? We also have a state-of-the-art addition to this atom probe, which is a furnace that is attached to an atom probe, where you can take these needles, you can anneal them at certain temperature. You can also, we had the capability of introducing any environment in the atom probe during annealing, and then study the material at that nanoscale during the destructive evaporation. So what we did was we made needles. there are these precipitates inside the aluminum, copper, manganese, zirconium alloy. We aligned one of the precipitates in orientation where we evaporate, we can collect that interface, okay, between the precipitate and the matrix. What we see is in the initial condition, when the material is not exposed to 300 or 350 degrees Celsius, there is no compositional partitioning of manganese or zirconium on this interface. You can see that if these maps show 0 to 1.5 atomic percent of manganese, but there is nothing over here. So manganese and zirconium are somewhere dissolved in the matrix. There is no piling up over here. But copper-rich precipitate and aluminum-rich matrix is seen over here. No manganese, zirconium. But when we now take this needle, we take it to the reaction chamber, we still have some of the precipitate left. So we are just focusing on one precipitate over here. We take it to the annealing chamber, anneal it at a higher temperature, and then we'll bring it back and analyze the same precipitate a little bit below this interface, and we'll see what happens. Okay. So we did that. We took that needle, we annealed it at different temperatures like 300 and 350 degrees Celsius for different amount of times, and we can come back and look at that same interface again. Okay. Before doing that, we can also take it to transmission electron microscopy and also look at the interface, right? The similar type of interfaces which lie below it, not exactly something which is on top, but we can look at some other interface as well. So what is happening is we start seeing some solute piling up at high temperatures. When we go to 300 and 350 degrees Celsius, we start seeing some solute atoms piling up. And when we take it to atom probe, we start seeing some zirconium and manganese piling up at that interface, which is shown by this graph, right? So there is something happening at 300 and 350 degrees Celsius, which was not clear in just TM analysis. When we take it to atom probe, do site-specific analysis, we see that there is zirconium and manganese that is piling up on the interface, which is probably responsible for this interface becoming stable at that high temperature, right? So this is a very small, unique example where an interface, which is a heterogeneous site, can allow these heavier atoms to come and segregate over there and create a metastability in the microstructure, right? So I just wanted to introduce this term metastability. Stable phase is going from theta prime to theta, which I showed initially, right? But it is not going. It is locked in this persistent metastability because zirconium and manganese are coming and piling up. Then it becomes difficult for this precipitate to dissolve. And what we get out of it is we are able to stabilize these precipitates to high temperatures, and we are able to get higher strength from the material at these temperatures, right? So metastability is something which normally we don't pay as much attention as it is needed. We talk about equilibrium phases, which is the final go-to stage where material likes to go. But there are a lot of metastable states that can be locked in that particular condition, and that allow us to gain very high strength and mechanical properties, a combination of those two. Okay. So once we talk about this metastable state, now I wanted to go towards more complicated microstructures and talk a little more about how we can explore this metastable state of material. Okay. Let's go towards more complicated materials. Over here, I want to talk about high entropy alloys or complex concentrated alloys. Over here, what are high entropy alloys? The definition, which is now pretty old, and some people might start changing it, but the traditional definition was if you add 5 or more elements together in 5+ atomic percent, there we can assume that the configurational entropy effect would be higher. And so the name comes from that, and they started calling it high entropy alloys because the entropy can be assumed to be high at these alloys. So what we are doing is we are adding a lot of elements together by what it means is, for example, we have A, B, C, D. We are trying to add them, and we are trying to go towards the center of a phase diagram. And we are, for example, if we add, we talk about steels, we talk about aluminum-based alloys. They are mostly 90–80% aluminum or iron. But in these alloys, there is no principal element. You start adding a lot of elements together, and we start talking about what will happen when the phase diagram, we are in the center of the phase diagram. Okay. So entropy is higher, but we cannot ignore the effect of enthalpy of mixing, because again, when we start talking about these binary ternary pairs, they would like to interact with each other. So enthalpy of mixing would have a role to play. Maybe at higher temperatures, we can talk about entropy being higher, but at lower temperature, enthalpy will also play a role. And that's something I will emphasize again. Okay, so I'll talk about a system which contains aluminum, titanium, cobalt, nickel, chromium, and iron as alloying additions. What we did was, or what we intend to do is, there was observation that when we add iron, nickel, chromium, and cobalt together, it forms a single solid solution, and that was intended to be stabilized by entropy effect. When you're adding these different elements with close atomic radius and near in that periodic table, they tend to form a disordered FCC. But what was seen was when we add aluminum and titanium to that mix, it distorts the FCC lattice and it starts forming precipitate. Okay, and so these were the key ingredient. Then when you add a small amount of aluminum and titanium, you start forming something different than FCC. And so what does it tells us that entropy itself is not sufficient when you add elements which have higher interaction with, for example, aluminum with cobalt has a high enthalpy of mixing, negative enthalpy with nickel, high enthalpy. When these elements are added, they have different atomic radii, they start overruling the entropy effect and they start forming some different phases. Okay, so with this preliminary introduction to these material, I just wanted to talk about how this different atoms like aluminum and titanium, when they are introduced in these high entropy alloys, they can start distorting and they can start forming different phases. Okay, and another thing to keep in mind is there would be different types of phases they can form. If you start adding small amount, there is one type of precipitate that can form. If you start adding a higher amount of aluminum, there is a different type of precipitate. So in a way, these alloys, high entropy alloys, are mix of so many things, and they allow you to create different types of microstructure. They are super duper saturated solid solutions. So if you provide the right type of initial energy, they can follow different transformation pathways. So that's why I just wanted to introduce these alloys, and we'll talk about how we can modify their phase transformation pathways, the states they can go if the conditions are changed. Okay, so let's talk about that. Okay. So how are we going to change the state of the material? How are we going to introduce the energy we want in the material? That could be the one of the way could be changing the temperature at which you are holding the material, thermal agitation. Other could be we can introduce a lot of defects in the material, right? By defects, I mean these are crystallographic defects. You have a material in which the atoms are aligned in a certain pattern, but you start deforming it. You start putting it under stress, you're putting under shock, you start introducing crystalline defects in the material, right? And one of the ways is by using a pin which can scratch the surface. We are introducing a lot of these defects in the material. This is one of the techniques I'm going to talk about is called friction stir, where you can stir the material and introduce a lot of defects in the material. But there are various types of defects. Cold rolling is one of the methods where you roll the material and reduce the thickness, and you can introduce a lot of defects in the material. So this is a stored energy, right? The defects are a form of energy in the material. Now, when you take the material to a high energy state, you allow it, and when you allow the diffusion to happen, it can choose different transformation pathways. It wants to reduce its energy, and it can choose various ways to reduce its energy, right? Okay, so when we talk about these high entropy alloys, what we have is a very highly supersaturated solid solution. We are in the center of the phase diagram. We have a lot of defects. What can happen is when you provide the right type of temperature and conditions, diffusion will happen. What can happen then is you can form phases which are coherent, which can be distributed within the matrix, phases which require higher energy sites like defects, grain boundaries, deformation bands that can form on these bands. Okay, so what can happen is, because you have opportunity to form different types of phases, the phases can choose where they prefer to form. Do they prefer to form within the matrix? Those are type of phases you can form. Phases that can form on the boundaries with the higher energy of formation. Then there are phases that can form on these dislocations and deformation bands. So in a way, it allows us to create a templated microstructure where different defect energies determine what type of phase will form there, right? So, and we have all the ingredients in that high energy alloy, which can form different phases. So it allows us to create those conditions where you can create a very complex microstructure with a different hierarchy of phases, okay? So then we decided to explore this a little more, but we didn't know what type of material we should use, right? So again, because high entropy alloys contain several elements, it becomes very difficult what type of material to choose. So we used a rapid prototyping technique, which is called LENS, laser engineered net shaping, where you can create, we can use the powders of different materials, and by varying the speed of the powder, we can create a graded microstructure. We can go from a certain composition to a different composition. Okay, for example, over here I'm changing aluminum. You can go from zero aluminum to a very high amount of aluminum, and you can study the whole microstructure from one end to other, right? Over here, the microstructure is changing as you go from one end to other. And so the idea was we want to find a composition where you have this opportunity to create different intermetallics, different phases, okay? And so I'll summarize what we found over here. We found that if you keep adding aluminum over here, you can go from single solid solution FCC to FCC plus L12 to FCC plus L12 plus B2, and then BCC plus B2. So we decided to have a certain amount of aluminum where you can have different transformations to different phases, and then started introducing our we started introducing defects and try to understand how the defects can influence the phase transformation pathways. Okay, so one of the first method we used was we will melt this alloy, which has about 8 atomic percent of aluminum. We will take it to high temperature. At high temperature, it is a certain microstructure that we form. Then we deform it, 90% cold rolling, take it back to high temperature, again, remove all the defects, and then take it to lower temperatures. Okay, we'll see what happens in this situation. Okay, quickly. When we deform and take it to high temperature, because the entropy effect is higher, we start seeing a single solid solution FCC phase. No defects, nicely recrystallized grains, FCC microstructure, randomly distributed all the atoms. Okay, this is atom proof result showing the atomic structure. Everything is randomly distributed. But when we take it to a lower temperature, for example, over here, 620 degrees Celsius, of course, it starts forming a new phase in the matrix. Strength increases. This is the mechanical flow stresses. They increase. We start forming these nicely well-distributed precipitates in the matrix. These are L12 phase. And on the boundaries, we start forming B2. Okay. So again, just to give you a glimpse what is happening in this particular situation where you remove all the defects and then took it to low temperature. But phase diagram, which is a thermodynamic calculation, shows that if you take it to 600 degrees Celsius, you should see FCC B2 and sigma. We don't see FCC B2 and sigma. We see FCC L12 and some B2 on the grain boundary, right? Then why are we not achieving what is dictated by the phase diagram over here? Okay. Okay, so before answering that, I just did another heat treatment where you did a cold rolling, and without removing all the defects, as in the first case, we took it to a lower temperature directly. Okay. In this situation, what we see is a very different microstructure. We see a fine scale grain structure, very small grains, which are about 500 nanometers. You start seeing B2 phase, another intermetallic. Within the grains, there is no other phase. And then you start seeing a sigma phase as well. Okay, so I'll show a little bit in the next slide quickly. So B2 is forming, some BCC is forming. Within the matrix also, you start seeing a very complicated microstructure. You have these clusters of aluminum atoms that are forming. So just give you a quick glimpse again. What we did was removed all the defects and then annealed at low temperature directly. Not a big difference, right? But completely different microstructures, right? Very complicated microstructure compared to what we saw initially. So summarizing it, if we remove all the defects and then take it to low temperature, we see a certain type of microstructure, mechanical properties increase. But then if you keep all the defects and then take it to a lower temperature and anneal, you start seeing a very different microstructure, and properties can be increased to a much higher value. Okay, so what is happening over here? The question is why we see different. Thermodynamics should determine a state of material. Why are we seeing two different types of microstructure? So just quickly summarizing what I talked about, there are two different things we talk about when we talk about materials and phase transition pathways. Onset driving force, that is the driving force of material to go from one state to other state, right? So there is a driving force for these precipitates to form B2, L12, sigma, BCC. There is a driving force, and driving force for B2 is higher. That's why thermodynamics determines this phase to be stable. But we more often forget that to reach to that state, there is a nucleation barrier. How these precipitates will form has to go through a energy cost in between. Okay, and so what we found that is L12 has the lowest nucleation barrier in FCC. B2 is higher. Okay, so if the conditions do not allow that long range diffusion, then material gets stuck in a metastable state. And L12 is a metastable phase at 600. So that's why we form L12 in a non-defective condition where there are less nucleation sites. L12 can form very easily, and B2 is more difficult, so it sticks on the grain boundaries. But when we deform the material and then take it to the right condition, B2 can easily form because there are a lot of defects, right? So we see a very different microstructure, and that is what we see, which is closer to equilibrium, okay? So quickly summarizing over here, what we did was we are able to modify the transmission path by just affecting the initial state. We cold roll. If we remove all the defects, we start forming this nicely precipitation strengthened material, FCC plus L12. L12 is a metastable state over here. But if we introduce a lot of defects and directly take it to low temperature, we take it closer to equilibrium. Okay. So I just wanted to leave you with this message that equilibrium is something which we more often talk about, but there are a lot of states material can exist, and that could be persistent metastable states, which can be more beneficial many times for the properties. And so defects are a knob that we can turn, and that can be used to introduce a different mode of phase transformation. That is what I've used in my research so far, and where initial state with different defective conditions can be applied to explore different phase transformation pathways. And over here, I presented the example of high entropy alloy, how this can be utilized. Okay. Thank you.
Thank you very much, Professor Gwalani. I really appreciate that wonderful talk. It's time now for us to head to our panel. So I'm going to introduce our panelist. And this is somebody who doesn't need introduction here. Alice is going to be joining me in the panel. Those of us who have been in ICANX know her very, very well. So let's jump into the panel. Alice, do you have any questions?
Martin. Yeah, Martin, maybe I go the first. Yeah.
Sure.
So as so lucky to hear these two wonderful talks, very inspiring. Actually, Charlie, you know, innovation chat box, you know, the chat was very impressive. Yeah, because I'm teaching a class for the innovation, how to, you know, teach students to do this job. I think this is very useful and helpful. Thank you very much for providing this. And Bharat, Bharat, right?
Yeah, Bharat.
Yeah, try to pronounce your name first. Yeah, so you know, for this kind of, I first see the title of the talk. I think should be like a transformer. You know, yeah, for the matter to something. Yeah, but I listened to your talk. I feel quite surprised because you know started out bulky, you know, materials, but to try to, you know, kind of analysis in the micro nano structures, try to, you know, modify all this. I have a question for you. Is that possible? You know, you have so many different elements, you know, kind of dop and all this thing. Is that possible to use the AI algorithm to, you know, simulation or do some, you know, kind of calculation first?
Yes, definitely it is. And there is a lot of ongoing work on that front as well. You know, in binary and ternary, a lot of work I showed, for example, was thermodynamic based simulation. And there is a lot of history of research in binary and ternary alloys. So a lot of these databases have been developed by experiment, and then they tweak the models. But when you go towards 4 and 5 element systems, first of all, first principle calculations become very computational extensive. So that we have to understand that, you know, doing DFT like calculations become very difficult, and you can only have a 200 to 500 to, you know, a limited amount of atoms that you are simulating. So if you have 5 elements, so each entity becomes very small. So their influence is underestimated. So DFT-based calculations are still open research. People are still working extensively on that. But thermodynamic-based, as we create a lot of literature, as we have so many papers now, they have been tweaking the thermodynamic databases and creating more and more real predictions of what type of microstructures you can expect. And again, I talked a lot about metastability, right? A lot of these models talk about equilibrium. So that is another active area of research, how you can determine or predict metastability. So again, there is a lot of work going on in that direction as well.
Okay, cool. Yeah, Martin, maybe I have another questions to follow up. Yeah, I saw in the last slides you mentioned that the defect sometimes can be helpful, right? Yeah. So yes, the thing is, normally we try to remove all this defect, right? Sometimes you think is you to, can be useful this, how to balance this, how to, take some advantage of all this.
Yeah, I think it is well said. I forgot the person's name, but our defects is what makes us interesting, right? And the materials behave similarly. I mean, all types of strengthening mechanisms we talk about in material are due to the presence of defects, right? If we talk about Hall page, grain boundaries, the smaller the grain structure, the stronger the material is. That's what allows to restrict that plastic deformation, and that's what makes it stronger, right? So that's why in materials specifically, defects are not bad. First of all, whenever we use a lot of our material, they are in a cold rolled state, which makes them stronger, right? So defects can increase the strength of the material. And the way I wanted to talk about it was, if you have these defected microstructures, right? These are high energy states of material. They want to reduce the energy of the material. So the place where you take the material by introducing the amount of defects you do, the type of defects you do, can determine where it wants to go now. Okay, end state is well known. Okay, equilibrium is well known, but most times equilibrium is so much far away that it will never reach there. It will get stuck in a metastable state, and metastability will be determined by where you take the material initially, right? It is a very high energy state. So you can determine where you want to persistently hold the material by introducing the amount and type of defects you have, right? So that's a knob you can always turn.
Okay, I see. Yeah, that's also a challenge. Okay, Martin, may I ask a question for Charlie? Yeah.
Oh, go ahead.
Okay, yeah, Charlie. So I saw that paper Dynamics, many times. I think, yeah, in the group we always study that because it's, the material was so simple, everyone can have it, can get on hand to do something. I saw you still, did a bunch of this work and can already be a series, right? Yeah, a platform. So you use that in Africa to detect some, something. So maybe this question is not being asked oftenly, but I have to ask that. Yeah. So this very simple, you deliver to anywhere, to any person, to any people. They can use that on hand and even can, take photos or something like this. But how about the waste? So once, yeah, this was a single use, right? Yeah. How about the waste?
That's right. Yeah, waste is difficult to get around, and I think that's one of the benefits of using paper as the main material of the device is that it will take a while, but is more likely to degrade than something that's made predominantly of plastic. If burning waste is the main mechanism of removing it, then it's going to produce less sort of toxic byproducts than burning plastic will. But yeah, I mean, I think one of the challenges is, they have to be single use. When you're putting a patient-derived sample on it, blood, saliva, urine, whatever, that's a biohazard. And so that's going to be, after the test is being used, that's now medical waste that needs to be disposed of. And so I think, moving towards using materials that are more environmentally friendly, or at least as environmentally friendly as we can have at this point in time, that's a target to strive for. And I also think, more directly addressing that by now trying to identify or make materials that have the properties that would make them less likely to to generate piles and piles and piles of waste, and then use that as the next platform for developing these kinds of devices. That's another area of research that I think people should be moving towards, if not now, then soon.
Okay, super. So can it down to 1 cent a piece? Cheaper like that?
I don't control that. I mean, that's that I would love for that to be the case all the way across the board. Making things is really expensive, and you have to make a lot of them. Thinking glucose test strip kinds of devices in order for production costs to get so low that consumer price would be around a cent. It's hard.
Okay, I see. Yeah. So thank you so much.
So let me jump in here, and I want to start with a very high level kind of question on this idea of transformation and translation. If you look at both your talks, it's like you're two different fields going in opposite directions. Charlie, you start with paper and you build simple devices, but as you go up, the work gets very complicated, right? You have people to worry about, you have quality control to worry about, you have defects in terms of error rates to manage. On the other hand, Bharat, you're starting with very high-end equipment, very expensive technology. By the end of the day, you're looking at something really, really simple. You're looking at defects, and you're looking at atomic to atom interaction, cohesive energy density, basically. So you're taking something like steel and taking the journey to understand that complexity. By necessity, you have to go simple. You have to even go to the atom probe to start mapping single atoms. And my question for both of you, because the reason I wanted to see both of you on the same platform is the contrast in your work, yet all of you are leaning on practicality. On one side, Charlie, you're doing diagnostics to solve a very simple problem. How do we know we are sick, right? But you're looking at very complex systems to try and understand how do we make strong materials. And for both of you, the question I have for you is, as you build these disciplines, where do you see information being shared across both fields, so that Bharat, you've seen from the steel level and coming down to the single atom level. Charlie, you start at the bottom and you're building up. How do you communicate information so that you can help very, very similar disciplines? How do you transform metallurgists and people working in diagnostics so they can talk? The question is yours.
Sure. So I think this is one of the great, sort of purposes of being an academic scientist, is being sort of compelled and obligated to share. Is something that a lot of companies don't feel the pressure to do, hiding, with their own intellectual property and their own art. We don't want to have that. I want to share. I don't want to hide the things that we do with others. I want to have the things that we learn be disseminated as broadly and as clearly as possible, so that way anybody else around the world can use what we've learned to make their next steps, because we are relying on what we're learning from the work being done by others. It's not an isolated place to be. It's a very, whether it's direct or not, it's a community, and we want to make sure that we're operating within that. And so yeah, I want, through places like this, through this series, this is an amazing place to reach out to people and to share our methods. And so that's kind of the hope. Maybe something we overlook gets taken up by somebody else and they run with it, or maybe the same idea is taken up by multiple groups around the globe at the same time, all to try to converge on helping people. I think that really comes from needing to and desiring to share.
Yeah, and I just want to add, because Charlie mentioned very nicely about why should I care, and the theme of the talk was how to take the science from labs to people, right? And I didn't talk a lot about that. But actually, the theme is similar in all sorts of scientific fields. We really want to answer that question: why should anybody who is not in my field care about it, right? So I talked about aluminum copper, for example, over there, and microstructures and properties. But how would it influence anybody in my neighborhood, for example? Things which we do in our lab, again, because this is a long scale problem and it takes sometimes tens of years to commercialize a material of that scale, because we are going to employ it in cars and planes and rockets and things like that. So it's not, it cannot be certified within a year or two. It takes longer time than some other things. But again, the process is similar. The process it has to go through, the material has to go through, is very similar. You have to do the proof of concept work, and then you have to go from TRL 1, 2, 3, 4, 5, 6, 7, and then it goes towards commercialization, and then some companies will start manufacturing it. So I think the whole sense about doing this in our lab, trying to show the proof of concept, doing all the thorough microstructural characterization to show the proof of concept and be sure that it will do the way I'm telling you to do it would. And then it gives confidence to a manufacturer that, okay, this has been well published and talked about and people accept it. Then they go towards manufacturing it. So I think the whole process is very similar. So that's something where I think, a scientific field, that's how it would go through all these stages. Another place where I think Charlie and I definitely can interact, and I think a lot of us interdisciplinary researchers do interact, is their need and our solution, our need and their solution. I think materials behave, everything is made up of atoms, right? And at some point, we all will interact with their atoms and they will interact with my atoms, right? By my atoms, these are metal atoms, and by their atoms, these are blood cells or something like that, right? So we have to find that compatibility. We have to find that the response of material that it would have in those kind of environments. And I think that's where a lot of us, which are working in interdisciplinary fields, interact. They talk about the environment, they talk about the things which they are going to put on the material I'm going to develop, and then I talk about how it would respond. Would it be able to sustain it? Would it corrode? Would it go to different phases you don't want? So I think that's where a lot of us interact, and we talk about what is happening at an atomic scale.
But actually, if I can jump in a little bit, you talk about high entropy solid solutions, right? And in the craziest of the world, nobody's going to look at high entropy solid solution and look at blood and put them on the same plate. But if you seriously think about it, blood is a very complex fluid. It's made up of many, many independent components. And at the end of the day, both the solid solution you're working on and blood are going to respect thermodynamics. So is there a place for us to start cross-talking between the biological world and the materials world by looking at fundamental thermodynamics to understand, if I can see how you're handling high entropy alloy, can I then take that to Charlie and say, hey, Charlie, this is how we can handle blood? Is there an underlying principle? Because at the end of the day, blood doesn't care, it's blood. It's mixing. Charlie wants the things to be completely mixed, right? You in like high entropy alloy.
Separations take energy, yeah.
Right? And Charlie wants to make sure that I can take the hematocrit this way and leave the cells here. For in your case, when you're adding the manganese and all these, you're trying to manage that miscibility. So at the end of the day, it's the same kind of problem. But in the craziest world, we never, ever would put a metallurgist and a bioanalytical physicist in the same room and ask them to solve the same problem. But if you look at cognitive diversity, when we think about dissimilarity, how dissimilar ways of thinking bring the best solution, isn't that what we should be doing? Isn't that what we should be pushing ourselves to get out of our comfort zone, to be able to provide solutions in areas that we have no clue, we have no background in, but we have the principles that can be useful and applied in that. Charlie, do you see an opportunity in that, or am I going too far?
No, I mean, absolutely. I mean, I think this is, you're speaking my language here. I mean, I think that there's when you get too far in your own subfield and you're surrounded by people that do what you do and think how you think, it becomes an echo chamber where you have a very difficult time sort of getting outside and thinking outside and doing outside of what you do. And I think that there's a massive benefit to having people from different fields, different backgrounds come together and to share in this way, so that way we can learn and take and borrow and then properly cite the work that's being done by other fields to do bigger and better things. There's most of what I've been able to do in my lab has come from work done by others, not necessarily in the analytical community, right? I mean, we're talking, fluid flow, biomicrofluidics. That's not what we do. We leverage that. We're talking, we borrow a lot from how things flow through, like soil, because that's a porous matrix too. I mean, there's just so much that we can learn from people that have expertise in other areas. I think we should be seeking that out specifically.
I couldn't agree more, yeah, it's really necessary to put the different people together, inspire each other. And especially, many times, you think about that, even though different material, but, you're fighting the same target, right? Yeah, you're fighting the same target to make a better life. For like Martin just a suggestion, which I'm really looking for, because my leg was broken, yeah, this summer. So we do need something. Yeah, we do need something with very strong stiffness, like a lawyer. We do need it can, communicate and nursing all this blood and bio tissues. This was, yeah, what we exactly looking for. I think this can be, kind of people go together, to think about something out of your mind.
Yeah, and I think that really big problems can't be addressed by individuals in a silo. I think you have to collaborate broadly, bring in others with disparate areas of expertise. It's the way of the world, and I think it's the way that science should go as well.
Great, yeah.
So I want to thank both of you very much for gracing the TNT talk today, transformation and translation.
TNT.
Yes, and then I like the fact that both of you come from a very, very practical point of view and dealing with simplicity and complexity at the same time, and really embracing that question of how do I understand such a complex system as a global health management and making it affordable by using very simple tools, or understanding the strength of a steel bar or a steel construction, metallic construction, using a defect of all things. And normally when we hear defects, we run away, right? But the ability to bring that science from a very simple point to a large scale is really, really appreciated. And we hope that those who are listening to us today will be encouraged to look beyond the confines of their discipline and think across the fields to address real problems, not just do science for the sake of doing it. And for the sake of time, we are exactly at half of the hour. I wanted to say thank you to all of you. And if we're in person, we would present these certificates to you. But we want to thank you for gracing our Young Scientist series, Volume 32. Charlie, we'll be sending this to you electronically. And then Bharat, we're going to be sending you one too. And we want to say thank you very much. And for everybody else who's listening to us, we want to invite you next week for this Chinese language, a Young Scientist series on optoelectronics, where our speakers are going to be Yanting Wan, Dingding Ren, and we'll have Shonglui Zhao, and they'll be moderated by Haiding Sang next week. We also want to invite you to our Friday series. Please join us at the same hour. With that, thank you very much. Thank you for joining us.
Okay, great. Yeah,
Yeah.
super talk.
So, gentlemen, I am very happy.
I didn't either.
Yeah, TNT talk.
TNT talk.
Yeah, that was
And
good.
by the way, we just
Martin
gave a
the
talk,
TNT
Charlie,
group.
Charlie and Bharat. Alice, we have about 15,000 people online, right?
Nice.
Yes, it is. Yeah,
Just on
we
the-
have
just on the
15,000
website.
people. Yeah.
Yep.
That's
I
awesome.
feel like Taylor Swift.
Don't worry.